Morris is a full member in the Computational and System Biology program at Sloan Kettering Institute and a co-director of the graduate program in Computational Biology and Medicine at Weill-Cornell. Previously, he held a CCAI chair through the Vector Institute for Artificial Intelligence, was a full professor at the University of Toronto, where he still holds courtesy appointments in molecular genetics and computer science, and an associate researcher at the Ontario Institute of Cancer Research. Quaid pursued graduate training and research in machine learning at the Gatsby Unit and obtained his PhD in computational neuroscience from Massachusetts Institute of Technology. The Morris Lab uses machine learning and artificial intelligence to do biomedical research, focusing on cancer genomics, gene regulation, and clinical informatics. So it's a pleasure to welcome Dr. Morris. Thank you very much.
Yeah, thank you for that introduction. There's some familiar faces in the audience, so Aziz might be somewhat familiar with this work. And so today I'm going to talk to you about two projects in my lab, and I'm going to focus, oh, here we go. Let's get this. I'm going to focus primarily on cancer genome sequencing and somatic mutations identified through that process. And we're going to be talking primarily about somatic single-base substitutions. These are the most common mutations that you see in cancers, and somatic just means that they are in the cancer. They weren't present in the germline of the patient whose tumor was sequenced. And generally speaking, we're going to assume that we're sequencing a cancer cell population that's heterogeneous genetically. So we'll get information not only about the mutations, but the extent of the mutations within that cancer population. So the assumption here is that all this is happening ahead of time. And the two projects I'm going to talk about is one which is a bit more of a translational project, and it's about the clinical deployment of tumor type classification. And this is joint work with Mike Berger's lab at the Memorial Sloan Kettering Institute of Cancer, Memorial Sloan Kettering Cancer Center. And it was with a joint student, Madison Darmfall, who just recently graduated. And this work is published, but recently published. It's not like anyone reads the literature these days anyways. But the other thing is, the other project I'm going to talk about is Damuda, which is unpublished work on using mutation signatures to have some understanding about DNA damage repair deficits in cancer. Okay, so why do we care about cancer type classification? So when you unfortunately have a cancer diagnosis, in most cases, the tumor type is relatively easy to determine because a lot of it's determined by the tissue in which the tumor occurs. But for 3% of patients, they appear with the cancers of unknown primary, meaning that the tumor that gets detected is not the primary tumor, it's a metastatic tumor. And in some cases, the primary tumor is either no longer detectable or it's unclear where it came from. And so those are patients for which the cancer type is unknown. There are other situations in which the cancer type is unknown. Often if patients show up with a second cancer after they've been treated for a first cancer, you don't know if it's a second primary or if it's a recurrence or metastasis from the initial cancer. And the reason this is important to assign tumor type is the treatments that are informed by primary sites can have much better outcomes. In particular, a lot of the targeted therapies that you see that are having transformative effects in some tumors are indicated, at least by the FDA, only in specific types of cancer. And so at Memorial Sloan Kettering, we're somewhat unique, though becoming less so in that most patients who come to our hospital have their tumor biopsy sequenced with a clinical sequencing panel. If we can biopsy the tumor, in many cases, they can be their tumor sequenced with an FDA-authorized clinical sequencing panel. And this was the first one that has been authorized, and we've been running it for about 10 years now. And and there's a whole pipeline by which patients consent. Many patients consent to research. We do the preparation, and we analyze their sequencing outcomes, and we generate a clinical report for the clinician. And our sequencing tumor type predictors dependent primarily on the clinical report. Okay, so people had previously tried to solve this problem. A few years ago, we developed a system we call Deep Tumor. And so what this system did is it looked at sequencing data from whole genome sequencing of tumors. So instead of just sequencing a targeted panel like the MSK-IMPACT, which targets about 300 to 500 cancer genes, we sequenced the entire tumor genome. And we had a relatively small training set. We had 3,000 tumors, and we achieved an accuracy of tumor type classification of about 91% at distinguishing 24 different tumor types. And this work was published. And at the same time, my colleague and my collaborator, Mike Berger's lab, they also developed a system based on the MSK-IMPACT data, which is what our system is on. They had a smaller training set, and they achieved much lower accuracy. And they used a random forest at the time. Now, one of the insights that we had from our work on the whole genome sequencing is that driver mutations, which are the mutations that are detected by our panel, we're not very good at distinguishing different tumor types. So obviously we were surprised. With the larger training set and the better clinical sequencing, we were excited to put this idea to the test. Now, in the context that we were working, we're putting this classifier in as part of, in some ways, the diagnostic pipeline. But it's very different to train a classifier to predict tumor type on research samples where there's a well-defined set of tumors that you're looking at, and all of the cohorts are approximately the same size. The distribution of patients that show up in Memorial Sloan Kettering Cancer Center is very highly skewed towards some cancer types. In particular, if you see here, this is the distribution. And so non-small cell lung cancer, that's our highest patient population, followed by breast cancer and colorectal cancer. And you can see there's a very long tail of very rare tumor types. And so if you look at the 42 most frequent cancer types in the patient cohort which form the basis of our training set, there's a twofold difference, 2 orders of magnitude difference in the size of the panels. And if you've ever done machine learning, in particular supervised classification, you can tell that these differences in class size or imbalanced class can cause a severe problem when you're doing the training. The other thing to realize is that we're only looking here at the 42 most common tumor types. There's actually hundreds of more, almost like we'll see in the following slide, about 2,000 more tumors that are just from rare cancers, where we have even fewer than 50 samples, patient samples in the training set. And so we're interested not only in a classifier that can distinguish between these 42 most common tumor types, but can also tell us whether or not the tumor type for this patient was not actually represented in the set, which is a much different problem than the problem that most people face when they do supervised learning within the cancer context. So here's the way our classifier works. So we take the input from the MSK-IMPACT. And so what our classifier looks at is actually essentially just the clinical report. We're not seeing anything in our classifier that the clinician doesn't see. And it's important to realize that what the clinician sees and the mutations that get signed out for the clinician report, they're actually a subset of the mutations that are detected by MSK-IMPACT. And in particular, a pathologist has to go through and certify each of the mutations that the clinician sees is a real mutation. So it's a somewhat limited report. And our training set contained 40,000 samples from 40,000 distinct patients. All these patients were consented for research, as most patients coming into Memorial Sloan Kettering are. And the way that Madison set up her feature set is she represented features at different levels of abstraction. So we have features that represent hotspots, which are commonly mutated positions in the cancer genome that reflect like cancer-associated mutations, often gain-of-function mutations in oncogenes. There's also, but she also represents whether or not a gene has been mutated at all, or whether or not the mutation for a gene is truncating, meaning that it's a loss-of-function mutation. the model we used was a hyperparameters ensemble of multilayer perceptrons. So multilayer perceptron is just your workhorse artificial neural network. I imagine in this course you've already seen things like this before. The way that these models work is they take as input some representation of the feature set, and that's 4,500 features. And what they output is they output a probability distribution over in this. I said 42 types. We actually only look at 38 types. A probability distribution over the 38 different cancer types. So what that says is just a vector. It's got 38 elements, and those elements are all positive, 0 positive, and they sum to 1. And so the classification is the tumor type with the highest probability, but we also have a number, which is the probability assigned by our system, that says what the confidence is in that classification. You can see over here, Madison is just showing 3 different confidences, but we have confidence in not every one. And this is the probability that's non-small cell lung cancer. Now, the last thing that we do, which is important for the setting in which we're working, is we have an ensemble of different multilayer perceptrons. What that means is we actually feed the input into 10 different neural networks that were trained slightly differently using something called a hyperparameter ensemble, which I will maybe explain in a moment. I haven't decided yet. but the output of the system is actually the average across all 10 of these networks. And that hyperparameter ensemble and averaging is very important in the clinical setting for reasons that I will explain over the next couple of slides. Okay. And the last thing that we can do is then we can analyze all these networks using something called Shapley value analysis to tell us what the features were that were important drivers of the prediction in each one of the tumor types, which ends up also being important because we're looking at a lot of rare cancers, and it's not clear what the individual important features are that are unique to those cancers. Okay, so just a better description of our input feature set. So just notice here that this bar plot here shows counts of the different types of features, and notice here that this is a log axis. So our feature counts range from basically between a couple features to all the way up to about 2,000 different features. And so we have the hotspot mutations. So these are frequently encountered mutations in cancer. So we individually indicate those as features, like whether or not you have a specific base change, a specific amino acid change at a specific location in the cancer gene that's often associated with gain of function. We have as I said, any mutation, the truncating mutations. In some cases, cancers depend very heavily on promoter mutations. For example, in the TERT gene, it is involved in immortalization of cells. We also have what are called copy number variations. So cancers, they undergo a lot of gains and losses of copies of individual genes. Some of those gains are whole chromosome or arm level gains, and some of those gains are just repeated copies of important cancer-associated genes like MYC. We also have mutation signatures, which I will explain actually in the second part of the talk, and a few other features that are strongly associated with individual tumors. Now, one thing to know is the features are extremely sparse. So on average, we have 5 substitutions per patient. We have a lot more CNAs, but our feature set, even though there's up to 4,500 features, very few of those are ones. Okay, so like I said, Madison selected carefully the 38 most common cancer types. She did a bit of merging. This is how we got from 42 down to 38. So there's a lot of cancers that are distinguished by clinicians, but aren't very distinguishable at genomic level. For example, cancers associated with HPV. So the cervical cancers and some of the anal cancers are very similar to one another. So from the original cohort, which was 4,800 patients, a number of them, there just was not enough tumor material to get good sequencing. We don't look at hematological cancers because these are solid tumors. The hematological cancers have a different panel. That gives us 42. Now, remember I told you that 3% of cancers are tumors of unknown primary? Well, that's this number right here. So there's 1,600 cancers in our data set that are of unknown primary or mixed. And then there's also, I told you about these rare cancers. These are tumor types that aren't represented in the classifier. We would still like to know that it's a rare cancer, but we don't know what type of cancer it is. So this is our final cohort that she split into training and test sets. So the test set was held aside and was not looked at until we needed to determine classification performance. And we're working primarily on the training set, but we do cross-validation on the training set for reasons that I'll explain in a moment. All right, so as I said, these are the cancers of unknown primary. These are excluded types. And then this is the training set count for the cancers included. And what we've done here is we just distinguish between the previous work, which was 2020 on the smaller data set using a random forest, and the new data set, which is GDD-ENS. And so, basically the rarer cancers weren't represented in the original classifier. Also, we added sarcomas, even though it's extremely hard cancer type to classify, because we wanted to be comprehensive. The good answer is that it actually, our classifier performs extremely well. So when we have whole genome sequencing, we have a lot of information about the tumor, and we were able to achieve this kind of high performance level. With GDD-ENS, we are able to achieve basically the same accuracy as whole classifiers trained on whole genome sequencing, but only on the high confidence cases. So as I said, the output of the classifier tells you the probability, but with which the classifier believes that that is the tumor type being indicated. And so if we threshold on that probability, and the threshold we use is 75% and call those high confidence, our accuracy on the high confidence samples is like essentially the same as whole genome sequencing, about 90%, right? And that's just kind of shown here with these bar plots. And then the last thing is we do this cross-validation, and the cross-validation is how we fit the different neural networks. So each neural network is fit to a slightly different training set, because each one is only looking at a 90% of the training set, and we're using the validation set, the other 10% of the training set, as a way of determining what are called hyperparameters of the neural network. And those hyperparameters are things like how many hidden layers to have in the neural network, what the learning rate is in the optimization algorithm. So each one of our 10 neural networks is slightly different from one another. But it turns out that just when you average together the outputs of ensemble like we're doing, especially if we're using a hyperparameter ensemble, which makes the neural networks a little bit distinguishable, a little bit more unique from one another, they tend to be well calibrated. And so what do I mean by calibrated? So when you have an output of the neural network or any supervised learning system that gives you a measure of confidence, you want your measure of confidence to be calibrated to how accurate the classification is. So for example, if our classifier says it's 75% confident that this is a breast cancer, you want it to be correct 75% of the time. And if it says it's 30% confident it's a classifier, this is a breast cancer, you want it to be correct 30% of the time, because then that number, which is its confidence is probability of output, it's actually a useful number to a clinician, because they can look at that and get an assessment of the confidence of the system. Now, a good thing about these hyperparameter ensembles is they're almost always well calibrated for free. Most clinical systems, there's an additional step where they calibrate to the patient population. Not all of them do it, but some of them do. And in particular, random forests tend to be not calibrated, and individual neural networks, as you can see over here on the right figure, they also tend to be not calibrated. So on the x-axis here, this is the threshold for confidence that I was telling you about, and this is the accuracy of the classification. And ideally, you want this line here to be at the y equals x line. And you can see the average of the ensemble is better or closer to that than the average of the single neural networks. If I showed you a random forest, it would be even worse, more poorly calibrated. Right, and so as I said before, our accuracy is about 93%, and that's for the 72% of patients that are above the 75% confidence threshold. Okay, all right, and so you can take these neural networks, you can do something called Shapley value analysis, and that Shapley value analysis can tell you what features are important in the classification. And when Madison did this, for features where it was known what the important factors were, like colorectal cancers. The vast majority of colorectal cancers have an APC mutation. That's often the first mutation in colorectal cancers. Melanomas are characterized by a very specific type of mutation. It's a CC to TT mutation. That mutation is caused by damage to the DNA due to UV light. And you can see here that's the feature that's most important in that classification. And overall, you can see the relative proportions of the importance of different features. The mutations are very highly important across all cancers. One of the things that we found, which was a bit surprising for us, was that these copy number variations are very important in the classification of the cancers, particularly in the more rare cancers. So if you read our paper, we've done this Shapley value analysis for all 38 different types of cancers, and you can see the important factors in determining the cancer for each. Okay, and so I told you that there's about 1,300 cancers where, at least in our initial cohort, where our classifier can't predict what cancer type it is because it's just not represented in the set of 38. Well, those cancers, we can still detect them because for many cases, they're below the confidence threshold. So what I'm showing here is kind of a reverse cumulative plot that tells you the proportion of patients for cancers that are represented at different confidence levels, and the cancers that are not represented, these excluded types at different confidence levels. And you can see that like most of the cancers that are rare, which is so 66% of them are below our high confidence threshold. So we would never classify those cancers confidently, which is good because we can't because they're not in our training set. And these represent, as I said, a very small proportion. Now, if we look at that other third of cancers that are above the high confidence threshold that we are confidently assigning to a category, they are misclassifications, but in 2/3 of the cases, we actually recover the correct organ system. So, we're saying that they're a type of like essentially colorectal cancer, but a rarer type that has its own category. Now, the important thing about our output of our classifier being calibrated so you can interpret the probability as actually a probability of success is that we can then just trivially incorporate other clinical data without retraining the system. So, at the point of diagnosis, not only do you have this, not only do you have the tumor sequencing, but you have a lot of other information. And the most clear information is what the site of biopsy is, where you took the tumor sample. And so different types of cancers exhibit what's called organotropism, which means that they metastasize preferentially to different tissues. So that biopsy site gives you additional information that you can use to determine cancer type. All cancers undergo histology, meaning the pathologist looks at like a picture of the cells in the cancer and assigns the cells to a specific type. And the major types are adenocarcinomas or squamous cells. And so that also gives you important information. And then clinical history can also give you information about what tumor type someone has. So if you can express all those things as a probability distribution over the 38 different cancer types, and in most cases you can trivially integrate it. And what Madison did is she called this an adaptable prior, but this is, within the field of machine learning, this is just a modification of technique called naive Bayes. So we can trivially incorporate that. And here's a real example where the genomic data preferred bladder cancer, but there was a small chance that it was head and neck squamous cell carcinoma. But then the site of metastasis and the histology strongly favored this type of cancer. And so when you took this information and you connected it and you multiplied it all together and renormalized in the adaptive prior, there was a strong preference for this cancer type, which was correct in this case. And in general, what we see is that we see this is a complicated figure, but it just tells you, the arrows tell you the direction from the correct prediction, incorrect prediction, high confidence, and low confidence. And you can see the sort of most strongly, the biggest arrow here is from the low confidence to high confidence areas. And this overall, when we incorporated this additional clinical data that was always available at point of diagnosis, we got a 4% overall increase in accuracy. But the most important thing is we got an 8% increase in number of high confidence patients. So we went from 72% of patients that we could diagnose to 80% of patients we could diagnose with some confidence. I won't go into this. This is how we derive the adaptable prior. You can look at the paper. Basically, it's naive Bayes. Essentially, well, maybe I will. So if what our GDD-ENS system essentially telling you is a conditional probability distribution. So conditioned on the DNA mutations that are available for the patient, what GDD-ENS gives you is a probability distribution or a cancer type. And so you can use Bayes' rule with this conditional distribution to turn it into a probability of DNA given cancer type times the probability of cancer type. And this is just your prior over cancer type. Well, you can replace that prior with a different one that you get from the clinical data. And then if you renormalize, you get out a new conditional distribution that incorporates the new prior. And you can show that it's like I said, it's naive Bayes. All right, and so we started this. The main motivation for doing this is to help with the diagnosis of cancers of unknown primary. The other motivation for doing this is it provides like some backup to clinical decision making, and it also helps us to annotate our patient, the clinical records of our patient if it's harder to automatically derive tumor type from the clinician notes. But for the cancers of unknown primary, what we found is if we applied it to that patient set of 1,600 patients, we found a 2- to 4-fold increase in the number of cancer of unknown primary patients matched to targeted therapies. So, as I said before, one of the main reasons that you want to be able to diagnose or assign tumor type to the tumor is that you can match it to an FDA-approved targeted therapy for that cancer type. If you have a cancer of unknown primary, there's no clear match there. And in fact, there are some therapies that are kind of cancer type independent, but there's very few of them. So in our data set, in our cohort, about half of the patients had a targetable alteration. So it's a mutation that would be affected by a targeted therapy. And and these are the predicted cancer types in our data set that are the output here. And so when there was a targetable alteration among those 800 patients, about half the time we could assign it a cancer type that was for which there was an FDA-approved therapy for that targetable alteration. The biggest one was the KRAS alterations in non-small cell lung cancer. The other ones were like Herceptin. So these are HER2 positive cancers right here, and so that we can assign therapies to 50% of the cancer of unknown primary patients with targetable alterations. The next thing we can do is we can expand this to other assays, and we've done that. It's in theory it's easy to do because there's a lot of overlap in the gene sets that are targeted. In practice, it's a lot harder to do because everybody processes their targeted assay differently. So the MSK-IMPACT, at least the version that we looked at, there's 341 genes that are targeted by the sequencing assay. These are the 4 other largest cohorts of patients with the target assay. Foundation 1 is by far the largest, and you can see there's a relatively large overlap in the genes that are targeted. And the way in which we did this analysis is we restricted our system to only look at the genes in the overlap. And when we did that without any retraining, we found that we were able to kind of maintain the high level of accuracy. We were able to maintain the proportion of patients that were in the high confidence area, and we were able to basically maintain the accuracy. Only seeing a drop of 5% when we go down to 68% overlap, which is great. What's not great is that each of these panels has a different way they process the data, and each of these panels reports different features. So one of the important things about the MSK-IMPACT panel that showed up in our Shapley value analysis was the importance of copy number alterations in doing this classification. And most of these panels are either not reporting copy number alterations or only reporting targeted alterations or not arm level alterations. So in theory, if we could take their data and reprocess it, we would get something similar to this performance. But in practice, we're not seeing that if we just use the features that they have derived just out of the box. We can retrain systems if the cohorts are large enough, and Madison's tried that, and it does actually work pretty well. But like this performance is only achievable if we have the same processing. Okay, so in summary, what we've shown is that we can get high accuracy tumor type classification already using a routinely applied clinical test that works for 97% of patients that are coming into MSK. And this is for tumors with as low as 10% purity. And this is trivially what we call multimodal, meaning that we can incorporate other data trivially just without retraining by just using this adaptable prior mechanism, right? So to do this, unfortunately, we need tumor normal sequencing right now. You do normal sequencing so you can distinguish between somatic mutations that are mutations in the cancer versus germline mutations, which are just polymorphisms that you've never seen before in the databases. And the MSK-IMPACT analysis pipeline is very important because we need these arm level calls and these specific copy number calls in order to get the performance that we're doing. And so and for us, this is a very important illustration because there's a lot of work right now to use other omics or other features of tumors as a way of calling tumor type. And what we're saying is we can already do high level and high accuracy tumor type classification without really changing anything from what we're doing at MSK. This is the first part of the talk. The second part's a bit shorter, but I can stop here and take questions if people have questions about this. So I don't see anything.
So I think actually we have a question on the chat which I can read out. So it's from the Vicky. She says, "Thanks for the great talk. Could you elaborate more on how GDD-ENS is currently integrated into MSK's clinical workflow? And when the model outputs low confidence predictions, such as for rare cancers, how do clinicians typically respond to them in practice?"
Okay, so I should be clear, we are not a clinically approved test. So and when I say it's implementing the workflow, when they get the clinical report, this is something that they can look at. But it's part of the clinical report. They get a clinical report back. They have all the signed out mutations, and then there's additional analysis that gets done on that clinical report. And ours is one of the features of that. And I think, I'm not sure if I have them in this talk. Let me just have a quick look. But in some versions of the talk, I actually can show you what it looks like. And the answer to your second question is we don't really know. We're not, people are using our pipeline, are using it because we're getting feedback from clinicians on whether this was helpful or not. Most people find it helpful. We don't have reporting on whether or not it's changed their mind about things. But Madison, this pipeline was kind of put into place about a year ago. And before Madison left, she looked at the number of patients been run through it, and there's a few thousand. And it seems like in some cases, it did change the clinical management, meaning that clinicians did choose therapies that were consistent with what the output of the classifier would provide. Now, whether that they used our classifier completely or they used other means, we don't know for sure. But we do know that when Madison was making the pipeline and people heard that we had a GDD-ENS was an improvement to GDD-RF, she got a couple of emails from clinicians saying, "Hey, this is a case. We're not really sure what to do here. Could you please tell us? We heard you have a new classifier." And then she gave them the output of her classifier, and they were happy with the output. So that's as much as I know. I wish I had more information, but that's where we're at.
Great. Thank you, Dr. Morris. I think we have another question from Laura, which can we take that now as well, or?
Yeah, of course.
Okay, great. So Laura's question is, in general, can you say that cancer type classification is more promising or helps more with selecting, for example, targeted cancer therapies, or is detecting therapeutic targets or biomarkers that may occur regardless of tumor origin more helpful on a large scale? Does your classification approach basically always include the latter?
I mean, so I think we're very interested in my lab in practical and being practical. Um, you know, if so, the question is, we can do tumor type classification, and then people have been trying to do this for a while. But then actually connecting tumor type classification to change in clinical practice is a lot harder. So, we did it. It seems to be affecting practice, and we're happy about that. Whether or not it helps detect like targetable biomarkers, um, I think it does. So one way of looking at what we're doing when you do the Shapley value analysis is we're kind of using data from across different tumor types to give us information about what the like cancer unique features are. And so by detecting these cancer unique features, the ones that are very like either they're like what the word is pathognomonic, they're like very specific to a specific cancer type. We are in essence finding things that are potentially targetable biomarkers that are specific to that cancer type. Now, we're not, we don't know how to target it. And most of the targetable biomarkers are things like, most of the things that we're finding in Cancer Unique are copy number or specific copy number alterations. So what you could do with this information is not clear to me, like right out of the box, but we do provide that. And there are papers that suggest, and like you can do the same sort of thing by just doing like what you would normally do in biostatistics, like a variety of statistical tests for looking for things that are overrepresented. But there is some like theory behind the idea that by like training this classification system, you're kind of making the best use of statistical power that you have. And we have data from like other work that we've done that suggests like the system learns something about or like a similar system learns something about the more common cancers that can help us in the classification of the rarer cancers. And we're already seeing that in that we get the organ system correct for 2/3 of the rare cancers that we try to classify that aren't represented by our system. So that tells us that we might be getting some power that would help us detect these predictive biomarkers. I hope that answers your question. All right. So I want to talk a little bit more broadly about what we're doing, and this is more of a research style talk than a clinical practice things. And so I call this part of the talk reading the cancer fossil record, and let me just quickly explain what that means. And so cancers evolve. Cancer evolution is a multi-stage, multi-phenotype process. And I'm sure many there's a number of precursor lesions that many cancers go through before they get like transformation into like a frank carcinoma. And in colon cancer, where these things are visible, this is pretty well worked out. And this is the sort of the genetic model of the development of colon cancer from Vogelstein. It's called the Vogelstein-Graham. And so when you are looking at the standard model of how carcinomas come to be is there's like a single cell that gives rise to the entire tumor. And so that single cell contains all the mutations that its ancestral cells accumulated both in the healthy colon and going through these different precancerous lesions. So in some sense, we can go backwards in time to sort of try to understand potentially what was going on during this multi-stage process. And so at the end of the day, we get a cancer genome, but the timing of these mutations, where they ended up in and how they ended up in the cancer genome, in some sense, that in some cases, that's actually possible to assign timing of the mutations. So you can say in what context those mutations occurred. Okay, and so let me just add this little cartoon that shows that, right? So if you imagine, so this type of mutation that I'm the mutations You can imagine the mutations you see in different cellular contexts are different. And I'll give you some justification for that in a moment. So if that's the case, then the mutations that are occurring at these different times within these different cellular contexts, within these different precursor lesions are different. And if we can assign each of these mutations to a different era, we might have a historical record of the cancer and I'm giving kind of funny names to each one of these things, but these are different eras that can be analyzed to understand what's going on at different stages of cancer evolution by reading this historical record. Okay, and so let me give you some idea of the type of information that these mutations provide. And so I skipped over this a little bit, but so when you sequence a cancer tumor, as I said, when we do the MSK-IMPACT sequencing, we see about 5 alterations, 5 mutations. It doesn't sound like a lot, but that's only because we're only looking at the coding region of a small number of cancer genes. When you sequence genome-wide, on average, you get about 3,000 mutations. So that's a lot of mutations, but the vast majority of them, they don't have really any function, meaning that they're occurring in areas that are non-coding, and they're occurring in areas that are likely not to be functional. So they don't tell you things about the gene function. They're not affecting gene function in any way, but they actually reflect the cell state, and they reflect the cell state in a couple of ways. So the first way that we know about is every mutation gives you a type. So the type is the type of substitution that you're seeing. So there's 6 possible substitutions: C to G, C to T, C to A, T to A, T to C, C to C. And whether or not a substitution occurs, in many cases, depends on the 5' nucleotide and the 3' nucleotides. So if you multiply 4 times 6 times 4, you get 96 different types of mutations. And if you just plot the histogram of the counts of those mutations in the cancer genome, you see that those spectra, these are called mutation spectra, they're distinct for different patients. So this is a mutation spectra of a breast cancer. And I can tell you, based on the height of these 4 lines here, these 4 bars, this is from an older woman. Because as I mentioned before, there's a type of mutation, CG to TG, that accumulates at a fairly constant rate as you age. I can tell you this set of mutations here. This is from a lung tumor, and this patient was a smoker because the C to A mutations, which are indicated by this red over here, they're the ones that are traditionally caused by tobacco mutagens. Also, this tumor experienced what's called an APOBEC-associated mutation process. And so APOBEC is part of your innate immune system in each one of your cells, and sometimes that becomes dysregulated in cancer and causes a whole bunch of mutations. And mutations that it causes are generally correspond to these 4 bars right here. Now I know all of this because about 15 years ago, someone went through at the Sanger in Peter Campbell's lab, Ludwig Alexandrov and a number of other people, took the thousands of cancers that have been sequenced and looked for patterns of co-occurrence of mutations. And these patterns of co-occurrence of mutations, like these 4 mutations I showed here, the C to A mutations, these 4 bars here, they detected through non-negative matrix factorization. They call these patterns mutation signatures. Right? And so now these mutation signatures, they exist in a database called COSMIC, and you can just go and look them up. And so some of the signatures, it was clear immediately what those meant. So these C to A mutations, it wasn't known at the time that tobacco mutagens caused these, but these C to A mutations, they were in lung cancer and liver cancer and head and neck cancer, and they were primarily in smokers. So there's a pretty obvious causal factor there that's then been worked out. These CG to TG mutations, I mean, it was already known that these mutations are pretty common. So it was no surprise that we would see these types of things. Right. And so when they initially defined about 30 different patterns of mutation signatures. Since then, this has been revised a number of times, and I'll show you that. But what you can do with these mutation signature patterns then is you can take this mutation spectra and break it up or deconvolve it into the contributions of each of these mutation signatures. And so here's the few signatures that I told you about already. So I told you about APOBEC. This is the smoking signature, the C to A's over here. And then, one of the fun things about this analysis is there was about half the signatures were what they call signatures of unknown etiology, meaning they didn't really know what they did, but they were common patterns of mutations. And that's one of the signatures here. And so you can estimate the exposures or the proportion of mutations that are due to each one of these signature types. And then you can also combine that. I told you can time many of these mutations. So you combine timing with the mutation signatures to look at the change in mutation patterns over the evolution of the tumor. And so this is what we call a trajectory of mutation exposures. And this is for a type of cancer called CLL. It's a B cell leukemia. And so one of the interesting things about this is that this mutation pattern, SBS9, it reflects like a common somatic mutation mechanism that's just part of the normal development of B cells that occurs in response to the activation of the B cells. And then, the IgH locus, the immunoglobulin, the locus that produces antibodies, gets hypermutated to try to find like more specific antibodies for a target that's been detected by the B cells. So this mutation type is either a misregulated process of this type, or just simply reflects normal B cell function that's occurring early in the cancer evolution. As you can see, as the cancer evolves, this mutation source disappears and other mutation sources appear. And what we found is there's a change in mutation signature exposure more than 70% of cancers, meaning we can detect this evolution of change in the types of mutations that appear in a cancer. The problem with this observation in general is it's very challenging to interpret. And so what we've seen, I told you we started off with 30 signatures. Well, now we're up to about 200 signatures. And the more data we get, the more signatures that get generated. And, depending on how you want to fit this curve, what we're seeing is an exponential growth in the signature. So it doesn't give you a lot of information, ultimately, if you just indicate a whole bunch of different mutation patterns. And one of the sources of this growth is, in our view, the fact that the mutation signatures, they actually involve two separate processes. So I told you there's like 96 different types, right? And part of those 96 different types are the context in which the mutation occurs, right? What the 5' nucleotide is, what the base that gets the mutated is, and what the 3' nucleotide is. And then the other thing is, so that's that context, that's what the damage process sees, right? So some damage process comes along, it creates some damage to your DNA. Most of the time, the cell repairs that damage, and so it's invisible. You don't see a mutation, but sometimes the cell fails to repair that damage correctly, and that's when you see the substitution. But those processes are distinct because the different DNA damage repair processes can target different types of damage, and different types of damage are repaired by different processes depending on the cell state. So there's actually a two-step process that's not being modeled by the mutation signature analysis people are doing. There's the damage followed by the misrepair. Right? And so this observation originally came from Teresa P's lab at the NIH. Hopefully, she's senior, so she's still at the NIH. And it was published in 2021 in the Cell Systems. But what Kate wanted to do is we saw this and we thought we could do an interesting model of this type of process. And so what we did is Kate developed a statistical model that distinguishes between these two different processes. So there's this model contains a set of damage signatures. These are co-occurring patterns of damage that just consider the trinucleotide context. It also has a set of misrepair signatures. So these are the types of substitutions that you see. And so there's co-occurring types of substitutions. And then this model contains an activity for each of the damage signatures, and it also has what we call an interaction matrix, which tells you which damage gets repaired by which misrepair process, or misrepaired by which process. And it's just it's a complex statistical model. It has millions of parameters because this interaction matrix has to be fit separately, and we fit it to 20,000 cancer genomes using a variational inference technique called ADVI, which was invented by David Bly and his graduate students at Columbia. And so what we found when we fit these signatures is that the damage signatures are more tissue specific than this repair. And this is not unexpected. We expect the repair processes, each cell to essentially have the same access to the same set of repair processes. And there might be minor variations in their effectiveness in different cells that we would see as tissue specific misrepair. but we expected the damage to be that you would experience to be quite different. For example, not all tissues are exposed to UV light. Not all tissues have this APOBEC process that becomes important. Not all tissues are exposed to tobacco mutagens. So on the left here is just all the damage processes, and on the right here are the misrepair processes. And this is the proportion of samples with the active signature, and this is just where we see significant enrichment of that. And you can see that the damage signatures are much more sparse in their activity, and the misrepair signatures are much more common. And if you try to cluster based on damage or misrepair signatures, it's the damage signatures better reproduce the tissues. We also found that this way of analyzing mutation signatures better distinguishes between things that do the damaging, like APOBEC A, and you can see here in data from APOBEC A knockouts that you see a decrease in different damage signatures, but you see less of a change in the different types of misrepair signatures. Whereas if you look at our repair enzyme UNG, loss of this enzyme shifts the misrepair away from one misrepair process to another, and has less of an effect on the damage processes. And so our activities of these different processes, you can summarize them in a 24-element vector, and they are much better at predicting altered repair pathways in tumor samples than the activities of these cosmic signatures. And we use the 75 cosmic signatures. So even though we have like a smaller feature set, we actually see improvements. They look slight, but they're actually pretty large within the context of what the range of improvements that possible are, and they're significant. And if you look at the importance of the features, it's the damage signatures that are much more important. Sorry, it's the misrepair signatures that are much more important at predicting repair alterations. So in summary, we have a new interpretation of these cosmic signatures as modes of interaction between common damage and misrepair processes. We can split these things up, and it's better. We're better able to study misrepair or repair deficiencies in tumor samples as a result of this modeling that we've done. So I'd like to acknowledge the people who did the work and our collaborators. This is my lab as of I would say 2 or 3 years ago. Our collaborators on Demuda are Tyler Fennell and Karen Campbell. Our collaborators on GDD-ENS are Mike Berger and his lab. And the people who did the work, as I mentioned, are Madison and Caitlin. And Madison is now graduated. And there are, at least in this version of the lab, there are some other people who contributed to the work that I showed. And happy to take questions.
Wonderful. Thank you so much, Dr. Morris. That was really interesting. I see already Aziz has got his hand up, but I do have a question that was coming about GDD-ENS that we never got to. So I'll start with that one first. So Carlos asks, is GDD-ENS presumably, is this limited to cases where only whole genome sequencing data and other clinical data are available? How could it benefit from the incorporation of, let's say, RNA-seq or methylation profiling?
Right. Great question. So just to clarify, it's not whole genome sequenced. It's panel sequenced, meaning that we're only sequencing 300 or now MSK-IMPACT's up to 500 genes. So we're only sequencing a portion of the genome. It's not limited to whole genome sequencing. So incorporating things like RNA-seq, I mean, we think that that's trivial because all you need your RNA-seq classifier to give you a probability distribution over the same 38 cancer types. And if we had that, we can incorporate that. But the big question is what's the added value of doing RNA-seq? And so I think that this is why we highlight the fact that what our performance is in something that's already routinely available. So there's a lot of work on RNA tumor type classification based on RNA-seq. And in general, those things seem to work a little bit better than DNA-seq. But it's a bit hard to evaluate that because unlike DNA, where the mutations are forever and they stick around with the tumor when they metastasize, you can imagine that the RNA profile of a tumor reflects the microenvironment in which it lives, which is going to change a lot from the primary to the metastasis. So So we're a little bit still uncertain about what the added value of RNA-seq is above and beyond what we already have. And that's particularly important because doing RNA-seq in the vast majority of tumor types is not it's not clinical standard, right? You almost never have that data. You have to get in a research setting. You have it for some things like breast cancer, like Oncotype DX is often done in breast cancers, but it's much more rare in other tumor types.
Great. Thank you. So, Aziz, do you want to go next?
Sure, thanks. Great talk, Quaid. My question is about the 18 damage signatures and 6 misrepair signatures. Do they all correspond to something that is, annotated, or are they just arbitrarily or empirically chosen to be 18 and 6?
I mean, we choose it using like standard model selection techniques, like the reproducibility across different seeds. We use like various types of like Bayesian model selection criteria. You could probably add or subtract 2 or 3. But those are the criteria that we use. It's the same kind of criteria by which people come up with like 75 or 200 things.
So
Whether or not they have like defined etiology, I mean, some cases they do, some cases it's obvious. But like with the cosmic signatures, there are a bunch that don't. Let me stop sharing and you can see my face. There's a bunch that don't, and we're not really sure. Like some of them correspond to like transitions, some of them correspond to like transversions, some of them correspond to like more general patterns of substitutions. And so we're working on trying to interpret that, but it's a bit hard to say necessarily.
I ask because, for example, especially for colorectal cancer, there was a distinct damage pattern, but when you looked at the M, all of them were activated, so it wasn't as sparse. So I'm wondering if that has to do with the biology of colorectal cancer and it being so aggressive that there's a lot of mismatch repair mechanisms that are recruited, or if it has to do with the sparsity that was chosen for the mismatch repair annotations.
Yeah. So, I mean, let me share it. Let me show you that again. So like colorectal cancer is not alone, right? Esophageal cancer has that same pattern. Stomach cancer has that same pattern. Skin has the same pattern. This D3 right here, this is UV light. So some of these correspond to like a mismatch repair deficiency. So the thing is that often what you see when you see a loss of a DNA damage repair process or inefficient DNA damage repair, it reveals a new type of damage that you otherwise wouldn't see. Right? So like in general, you see a lot of these mismatch repair, these misrepair processes active across tissue types. And then here you have kind of damage specific or tissue type specific damage appearing. But like you can see that, this is pretty dense and this is pretty sparse, and many of these tissues are distinct in the same way that colorectal cancer is distinct.